Okay, so I would like explain about recombinant DNA technology. And recombinant DNA technology is the joining together of DNA molecules from two different species. The recombinant DNA molecule is inserted into the host organism to produce the new genetic combination that are of value to science, medicine, agriculture, and industry. Recombinant DNA technology compares altering genetic materials outside an organism to obtain NH and desert characteristics in living organism or as their product. This technology involves the insertion of DNA fragments from a variety of sources having a discernible gene sequence via a process vector. And we can see in here, we have application of recombinant DNA, such as vaccine, growth hormones, antibodies, vectors, and etc. And the process of recombinant DNA technology. The step one, isolation of genetic. The first and the internal step in the recombinant DNA technology is to isolate the dessert DNA in it is pure from free from other macromolecules. And step two, cutting the gene at the recognition site. The reaction enzyme play a major role in the determine the location at which the dessert gene is inserted into the vector genome. These reactions are called restriction enzyme discussion. And step three, applying the gene copies through polymerase chain reaction or PCR. It is a process of applying a single copy of DNA into thousands to millions of copies once the proper gene of interest has been cut using the reaction restriction enzyme. And step four, ligation of DNA molecules. In this step of ligation joining of the two species, a cut fragment of DNA and vector together with the help of the enzyme DNA ligase. And the last step is step 5, insertion of recombinant DNA into host. In this step, the recombinant DNA is introduced into the receipt host cell. This process is termed as transformation. After the insertion of the recombinant DNA into the host cell, it gets multiplied and expressed in the form of the manufactured protein under optimal condition. And we have the types of recombinant DNA too. The first is the endonucleases cut within the DNA strand. The endonucleases cut within the DNA strand, where the exoscleases remove the nucleotides from the ends of the strand. The restriction endonucleases are sequence species which are usually palindromes sequence and cut the DNA at specific point. And the last is the exonucleases, remove the nucleotides from the ends of the strand. And then this is the example of recombinant DNA, yeah. Food and agriculture, health and disease recombinant DNA for the improvement of human health, like gene therapies, production of antibodies, investigation of the drug's metabolism, development of vaccine, and recombinant hormones, Chinese medicine, medically important compounds in berry and in environment, phytoremediation and plant resistance development. Genetic engineering has been widely used for the detection and abortion of contaminants in drinking water and other sample, and energy applications for microorganisms, especially cyanobacteria mediate hydrogen production, which is environmental friendly energy sources. And in the left side, I input the pitch of recombinant DNA technology. Yeah, we can see the process, yeah, in here. Okay, the first treat for each DNA and plasmid with the restriction enzyme and DNA ligase. And the second is introduce the recombinant plasmid into cultural plant cell. And the third, regenerate new plant from cultural cell. Okay, I think it's enough for me. Thank you very much.